This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 390, recorded on May 20th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me right here in New York City, Dickson Despommier.
Hello, Vincent.
Where it is a lovely day.
Gorgeous
just a few thin clouds, 22 Celsius. Finally, we have a bit of spring, right, Dickson?
Exactly.
And you went fishing yesterday, right?
I did.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it is similarly gorgeous here, 72 Fahrenheit, nice clear blue sky, really, really lovely, and apparently not going to be that way for much longer.
What's happening? Snow?
Rain coming in. Fortunately, not snow.
You know, it snowed this week in Montreal.
Really?
Up in Canada,
Nice.
Yeah. Also joining us from North Central Florida, Rich Condit.
Hi, everybody.
Welcome back.
Thank you very much. We got 75 degrees here, muggy and overcast.
Muggy.
With rain all around.
Good.
Coming and going. Yeah, the seasons are flipping here. I mean,
You need
we're getting
rain.
into summer.
Need to have rain. But
So I'm leaving. I'm out of here.
where are you going? Are you going?
Well,
You're
I'm
going
see
to
you next week.
Pennsylvania, right?
Yeah.
All right. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How you been?
Good. The weather here is 69 Fahrenheit, 19 Celsius. It's partly cloudy, but it's nice, and it's supposed to be nice all weekend.
Wonderful.
It'll be gorgeous.
And of course, all the undergrads have left, right? So,
Oh, long since gone.
and they're
Yeah.
nice and quiet there.
Pretty much.
Yeah, this week has been graduation for us, right, Dickson? Can't wait to get rid of them all. Oh, no. I don't really mean that.
Not the way to put that.
Today we have two guests from the state of Washington, and specifically Redmond, Washington. They both are working at Microsoft Research, and one of them is the lead researcher on a project we're going to talk about today called Project Premonition. Ethan Jackson, welcome to TWiV.
Hello, nice to meet you all. It is there's a hurricane going on, and it's raining cats and dogs here.
Oh, my. Raining in Seattle?
It's actually very nice.
How odd.
It's actually very nice here.
That's just as we imagine it.
It's a little known secret that the sun does come out sometimes.
Also from Microsoft Research, a researcher, Jonathan Carlson, welcome.
Thank you.
And you guys both can look out the window, right?
Actually, we're sitting in a windowless conference room, but normally...
I'm sorry to hear that. We have a lovely view of the Hudson River here in New York City.
Definitely jealous. Definitely.
Now, today, you guys are here because I heard about Project Premonition a couple of months ago when I visited University of Pittsburgh, and I spoke with Jim Peepus, who's one of your collaborators.
Absolutely, yeah.
And he was telling me about this, and I said, do you think that they would like to talk about it on TWiV? And he said, oh, yeah, you should. They're really cool guys. So here we are, and we want to hear all about it. So first I want to hear just a bit about both of your backgrounds, because I assume neither of you are virologists, right?
I'm a sort of adjunct virologist.
You are. So what's your background, Jonathan?
So I was an aspiring virologist growing up, and got that door slammed in my face the first time I stepped in the lab and realized I couldn't do anything right there.
And it's a dangerous place if you do.
I had that experience and went all the way through graduate school.
Well, see, I was smart enough to switch fields between undergrad and graduate school.
That was a good move, yes.
Yeah, I switched over to computer science, figuring that if nothing else, I could recompile and try again.
And you could still go viral.
That's right. Exactly.
Where did you grow up?
I grew up in Oregon.
You went to undergrad there as well?
No, I went to undergrad at Dartmouth out in New Hampshire, and then back to Seattle to University of Washington for grad school.
And where is it that you stepped into a virology lab?
Well, I'm talking about undergraduate biology here. This is very basic.
Dartmouth.
Yes, I made it through my undergraduate degree in biology only by identifying very competent lab mates to a whole bunch.
I know a virologist at Dartmouth, Chuck Coles. I don't know if you know that name. Charles Coles. No, Charles Cole, sorry.
Cole.
One call.
And Elmer Pfefferkorn.
Elmer, yeah, did you know Elmer Pfefferkorn?
No.
Great
That's
teacher.
certainly a memorable name.
It is.
Yes.
Great teacher, too. Ethan, how about you? Where are you from?
Yeah, so my background is purely in computer science. My research has been on how you make software not fail, not crash, particularly in the context of kind of robots and what we call autonomous systems that do something interesting in the environment. My undergrad was at University of Pittsburgh, coincidentally. And then I went to grad school at Vanderbilt. So, I had the privilege to be around a lot of interesting biologists, actually. And it's always something that sort of stuck in the back of my mind, how and when should computer science intersect with biology? And I think that's something we're actually seeing in a whole bunch of different fields right now. Jonathan is a great example of that.
Absolutely.
Yeah.
So your background is also computer science?
That's right, yeah. And then a subfield that we call, in computer science, we call formal methods, which are basically ways to try to reason about a program kind of like it's a mathematical formula. And if you want to ask, will a program crash, you try to ask, is there a mathematical proof telling you that it can crash, or is there proof that it won't? And it's through these kind of methods that a lot of advances have been made to create software that can do something interesting without failing. And as we talk more about this project, that's something that will come up a lot. That when we do interesting things in the environment, it has to go right.
Right.
When did you go to Microsoft, Ethan? Right out of grad school?
That's right. Yeah. So I've been here for about 8 years now, and I joined. Right out of grad school. So maybe I should just speak briefly about Microsoft Research.
Sure.
We're about 1,000 PhD researchers worldwide. I think we're a pretty unique lab in that we in many ways have a university-like model. We have postdocs. I started here as a postdoc, and we do research all over the spectrum, really with an eye towards pushing the envelope. So that's how I ended up here, and that's why we can do these kind of really interesting projects.
And Jonathan, you also came there out of grad school?
Yep, I started back 8 years ago as well, and actually have been studying viral evolution at Microsoft.
Nice. The biological kind of viruses, right?
Yes, the interesting kind.
Kind that make you sick rather than crashes your computer.
Right.
Those might make you sick as well, just in different ways.
Yes, really.
Yeah, but when computer viruses steal money from your bank account, it makes you sick too.
So tell us what is Project Premonition and how it came to be.
Because we didn't see it coming.
But I had a feeling it would.
Perfect.
We're very corny here on TWiV. I'm sorry.
Yeah, well. You said you only have an hour for this?
Yeah, they have an out. They have a hard out.
They have a hard out.
Right. So Project Premonition started really in 2014, sort of in the middle of this, West African Ebola outbreak, where it really struck us how one of the viruses that were believed to be the rarest on the planet suddenly had such a major impact to the U.S. I mean, that was something that who would have guessed it. And coming from the computer science point of view, the technology point of view, we started to ask the question: is there a better way to see these sorts of unexpected events besides waiting for these outbreaks to happen? When we started to dig more into that problem, it showed that trying to get data, trying to make predictions about these outbreaks is really tough for, I would say, 3 reasons. One is that 60 to 75% of these sorts of events are zoonotic. So how do you predict that some funny virus evolving in a bat or a dromedary camel or a mouse is going to jump into the human population and cause disease? Another problem that's clearly hard is just the the time it takes to observe that an outbreak has started. We know from data from the WHO that this delay is about 2 weeks, right? And under many epidemiological models, 2 weeks is too long to stop a disease once you've noticed that that outbreak has occurred.
Of course, for Ebola, it was like 3 months.
Yeah, and then for Ebola, it was 3 months, and so societal and political factors can also have an impact on on when you can really detect an outbreak. And then I think the third statistic that's very striking is just what we learned from metagenomics, that if we take a sample and ask what sort of viruses are in there, we will just find a bunch of novel ones. In fact, a large proportion of the viruses we see will be novel. And many of the recent emerging infectious diseases have been novel viruses, coronaviruses in the case of MERS, and SARS. So when you put those three facts into a bag and you shake them up, it's a pretty challenging problem. And how do you make a prediction when you have all of these unknowns floating around? And that's where we started to think about, is there a way to just get a lot of blood samples from animals in the environment? What if you could do that at low cost, amortized low cost, in a high throughput way? Would it give you a way to see potential pathogens before they lead to a major outbreak, where you then later detect that outbreak? So sort of being in a tech company where we turn everything into a device, we turn everything into a service, we ask, can we turn a mosquito into a device? Because a mosquito is really good at flying around, finding an animal, and taking a blood sample from it. So that was the original premise, and we started to really dig into how people collect mosquitoes, how people analyze them, how people do metagenomic studies on blood-feeding arthropods, to ask, is the concept feasible to build a system that would go out and in a high-throughput way, amortized low-cost way, collect mosquitoes to provide a background information on the kind of possible pathogens moving through the animal populations in the environment. So that's a really brief summary. I don't want to ramble the whole time about. I'll let you ask some questions about it. But that's roughly Project Premonition. Can we do that? And at this stage, we've built a lot of interesting technologies towards this goal. So we can dig into those a bit more. So is there anything you want to add, Jonathan, to that?
Yeah, no, I mean, I'll say that this is, in some senses, not a new idea. People have tried to use mosquitoes to sample, for example, influenza, but it's never scaled because capturing mosquitoes is hard. Doing that analysis is hard, and nobody's really tried to scale it as far as we can tell.
So you've limited yourself to what viruses that are picked up by mosquitoes. Obviously, there are many more than that, but it's what you thought you could do, right? Is that the reason for that?
Right. Well, I mean, the goal of when we say a mosquito as a device, we really view it as something that can go out and collect about 2.5 microliters of blood.
Yeah.
And we view it as, of course, that there's a time constant to the data in that blood, because it will be digested. So from our point of view, if there is, what we hope is that viruses and pathogens that might be picked up by a mosquito that don't infect the mosquito could still be detected. Mm-hmm. That's the hope.
Like I said,
is there any precedent for that? Because people have, used, as you said, people have pursued this sort of concept before, and without all of the technology. Can people find non-arthropod-borne viruses in mosquitoes?
Yeah, I mean, there's a limited research in that. I mean, one of the best examples I know of is a paper from a decade or so ago where they were looking at avian farms in Asia, I believe it was, and capturing the mosquitoes, and they could identify influenza A in those samples. They, in that case, they showed that influenza was actually replicating in some of the mosquito cell lines, but it clearly wasn't transmitting, and they were only finding it in blood-fed mosquitoes, et cetera. And people have looked at this with some other viruses as well, because, one of the first questions when HIV came out or with Ebola is, could this be arthropod-borne? So the first thing you do is you inject arthropod abdomens with blood and see if it can replicate. And usually it doesn't replicate, and it stops there, but a side effect of a lot of these studies is that you find that the virus is still in the blood meal for a while until it gets digested.
Right.
Yes. Yeah. So just to add on to that, I mean, so blood meal analysis is commonly done with mosquitoes and other blood-feeding arthropods. And so what's clearly been established there is, for instance, host is detectable, even though that host DNA is not replicating in a mosquito. And as Jonathan mentioned, a lot of the studies that try to determine if a pathogen is mosquito-borne, they do these experiments where they intentionally put it in the mosquito, and then they test it at different points in time, or they allow it to feed on some sentinel and then evaluate whether that sentinel becomes infected. And so what we know from these studies is that there are time constants for detectability using different methods, whether it's PCR or something else. So what's different, though, is that those experiments have usually tried to answer the question, does a mosquito vectorize some disease? But as a side effect, as Jonathan just mentioned, they also tell you that you can detect these non-mosquito-borne diseases with some time constant before the nucleic acids are digested in the gut.
Mm-hmm. So can you tell us how the technology works?
So what was really interesting for us from a technological point of view, so let me just take one step back. So, out of all the blood-feeding arthropods we could think about, why mosquitoes? Or why blood-feeding arthropods in the first place? So, finding animals and getting blood samples from them, it has been done. HIV is a good example where there have been studies going out taking blood samples from chimpanzees to try to understand the evolution from SIV to HIV. But we know for all of getting blood samples from animals is complicated and expensive and slow. If we view mosquitoes as a proxy, they're one of the best studied arthropods, and there are technologies that we know work for trapping them, for catching them. However, those technologies are still pretty low throughput, though they are effective. The basic design is this. You typically take some kind of fan connected to a net, and the fan is an impeller. It's going to provide a suction. If an insect comes near this fan, it sucks it into a net. So that's the basic idea.
Right.
And then on top of that, you add some kind of lure. The basic lure for all blood-feeding arthropods is CO2. That's either placed around the trap in the form of dry ice or a canister of CO2. And this forms the basis of all of these systems, all of these trap designs. So you put one of these things out into the environment, you leave it there for 12 to 14 hours, CO2 is dispersed. The idea is bugs come in, they get sucked into a net, you come back the next day and you collect your net of bugs, and then you process them and figure out what's in them. So that's the basic technology. It was developed in the 1950s, 1960s, and that basic technology is still what's used today by most public health organizations and researchers. The major challenge with it is that it's inefficient in some important ways. So to illustrate this, if you would go out and place one of these traps, come back the next day, the first challenge you'll experience is that it's full of bugs.
Right.
and probably 95% of these bugs will not be mosquitoes. They will be, particularly if you add a UV light, which is also a general attractant for insects, it's going to be moths, might be biting midges, it might be flies, it'll be all sorts of things. So what you first will encounter is before you get to the interesting data, you take this back, you throw it in an ultra low for 10 minutes, you dump out the samples under a magnifying glass, and then you have a well-trained entomologist sort them. So that's the first bottleneck in this process, is getting the samples in a form where you can analyze them. And so that's the first thing we've started to ask, can we throw technology at this so that we eliminate that bottleneck? We get from the environment to analyzable samples faster. And this led to developing what I don't think it's boasting to say the world's most advanced mosquito trap. And so I'll tell you what this mosquito trap looks like and what it does. So the idea of this trap is to sort of be that entomologist and on the fly only catch insects that you're interested in. So it means that it has to in real time speciate them and then collect them if it's a species that it wants to collect. The way this trap works is instead of having this bag and a fan attached to it, it's this honeycomb design of these kind of smart cells. Every cell is an independent collector. I think this is a lesson from high throughput biology that when you put things in regular wells, it's nice. You can process it more easily. Mm-hmm. You get one sample per well. You can tag it and you can keep track of everything. So the design is basically that. It's this honeycomb of smart cells. When an insect flies into one of these smart cells, it crosses a sensor that the insect can't see. This sensor is an infrared light that beams from one side of the cell to the other. When it crosses this light, its wings essentially cast a kind of an invisible shadow on this infrared light. And it would turn out that research done at UC Riverside and a number of other places have shown that this, what they call wing beat frequency, wing beat pattern, is distinguishable, can be used to distinguish one species from another. So insect flies into one of these smart cells. In real time, it's crossing the sensor, casting a shadow on this infrared light, and it's performing a spectral analysis on this kind of shadow and then deciding whether it's something it wants to capture. If it does, it can actually actuate the cell and close it, trapping that particular sample in that particular cell.
How big is the cell?
A cell is about the opening is about a square inch, and it's about an inch and a half deep.
Okay.
And so the design we have has 64 of these cells covering a cylinder, so it's symmetric from all sides. 64 because when we catch mosquitoes in existing traps, people are usually happy if they would get like 60 to 80 mosquitoes. That's a good trapping day if you get that number. And 64 because computer scientists work in powers of 2.
Right.
So we round to the nearest power of 2. So when they enter one of these cells, it can detect them. It now closes that door. In addition, there's a whole array of environmental sensors that measure the environment at the moment that door closes. And when it does that, it then tags the time of collection along with all these all of this environmental data that's now associated with that specific specimen. So you can imagine you come back a day after putting this trap out, you have a number of these cells closed. Each closed cell has a single specimen in it. And now you have a bunch of metadata tagged along with that specimen. It gets carried along with it because the samples are separated.
Mm-hmm. So as computer scientists, your approach then to the whole problem is to build a bug tracker.
That's right. Yeah. That's right. Yeah. Right. And now you can imagine the sort of data that comes off of this. The data that comes off shows you what that insect sounded like. So if it's an insect you've never seen before, because this is very likely actually when we put these things in the field, we don't know the wing beat frequency of some random moth or some odd mosquito species that can't be raised in an insectary. We will start to get that data as you put these devices into the field, and that becomes a training set. If entomologists can morphologically speciate and say, this funny insect is this kind of mosquito or this kind of moth, we have now the data that matches that sample.
Right.
And now the system can learn, right? The trap can become more optimal, ignore things that are irrelevant, focus on things that are relevant.
So what is powering this trap? There must be a processor or something in it, right?
Yeah, that's a great question. So, again, from the technological point of view, I think low-cost capable microprocessors have innovated a lot of technology. I mean, you think about the Nest and how it sort of disrupted the thermostat. Who would have thought that's possible? If we look at these classical entomology traps, it's the same thing. They have not made use of these low-cost, quite capable microprocessors. So what's in our design is one of these processors. It costs about $10, but it can do a lot of stuff. It's low power, runs at 48 megahertz. Can do all of this real-time analysis, and is powered basically off of a lithium-ion cell phone battery. So it's pretty light. Power source is pretty lightweight, right? It's recharged by your classic kind of micro USB charging cable that you would charge your phone with. So that's the processor architecture, and it's really taking these these advances in computer science and trying to push it into field entomology to accomplish that.
And I guess you and your team have written the OS for this device, right?
That's right. Yeah. So we developed the device, the electronics, the mechanics, and the firmware, which is the software that runs on the device. Were all developed in-house here. That's right.
And that device that you're talking about at this point, I mean, we're going to get, I'm sure, to the ultimate goal of this, back to the ultimate goal of this, but this is really kind of at a prototype stage where you're trying to catalog all these wing beats and evolve the device to the point where you can build something that is just going to catch the mosquitoes that you want, right?
Right. So from my point of view, the moment it crosses the prototyping phase is the moment that it's in the field doing something useful. And
okay.
And at this point, we've done, I think, all reasonable tests we can do in the sort of a controlled environment. Controlled meaning of the laboratory, insectary, and greenhouse, where we are releasing, known species of mosquitoes, and then looking at the data and seeing, are they distinguishable by wing beat frequency? Is the trap able to catch them? So we've done this on some important species, Culex fasciitis, which is one of the major West Nile vectors, Anopheles stevensii, Indian malaria vector, and of course Aedes aegypti, which carries all sorts of nasty things. Most recently, Zika. And so in these laboratory and greenhouse studies, the device works pretty well. And what that tells us is the next step is to ruggedize it so it will survive the field, it will survive the humidity, the rainstorms, and put it out and have it start collecting data. I think realistically, if it collects the data that you need, then it's at the right place, because what I expect is that every habitat we put it in, it will encounter species that it has not encountered before. And so I suspect there will always be this kind of training phase that you acclimate it to an environment where you let it sort of catch a broad set of insects, and then you train it to recognize the ones you want to catch in that environment. That's sort of what we expect will happen.
Okay, so you don't envision an iteration of this project where you then take those data and let's say you want to look primarily at mosquito populations in northeastern Brazil, to pick a non-random spot, where you might then adapt this design and say, okay, we were using this $10 processor and this cell phone battery, but we really can narrow the problem down to use a $2 processor and a smaller battery because now we just want to catch this one focused set, and now you can put more of those out in the field.
Yeah. So I think algorithmically, the algorithms that are doing that differentiation to only collect Aedes aegypti in northeastern Brazil, they aren't going to change really.
Okay.
When you specialize it to just that case, what really changes is the conditions under which you tell this algorithm to close the door. So the really cool thing about these sort of software systems is that they can adapt very quickly. You don't need to redesign the hardware at that point. You just need to change their definition of what they're interested in, and that's something that can happen really on the fly. So
to speak.
Or on the mosquito, yeah.
Yes.
The mosquito is a fly.
It is a fly, yeah.
Okay, so it makes sense to keep this as a sort of a general purpose device then.
Right, and then it's really a parameter of the device to define how specialized you want it to be, and that's all a software setting. Yeah.
How do you get the mosquito next? You need to get the mosquito into a lab, I presume, right?
That's right. Yeah. So the design, if you can just imagine in your head, this kind of nicely arranged cylinder has these cells in a very regular spacing, and we did this so that it would be easy to extract the mosquitoes from them. I mean, from the device. You can easily imagine, you sort of drop it into some kind of extractor that sucks them all out. We haven't built that yet, but for laboratory tests, what we do is take this device, we flood it with CO2 to knock down the mosquitoes in this little cylinder. So concretely, you put it into a bucket, you screw the lid on, you flood it with CO2 for about 15 minutes, as opposed to throwing it into an ultra low, which temperature cycles all the mechanical parts, electronic parts at minus 80, which so we do something at room temperature. And then basically you just open each cell with a little tool, kind of hooks onto the side, opens the door, and you just pull out the sample with a pair of tweezers. That's the protocol right now.
Mm-hmm.
What size are these? I'm picturing something about the size of a coffee cup. Is that about right?
You mean for the whole device?
For the device that you would set outside.
Yeah. So the device is it's a bit bigger than that. I would say it's about 18 inches tall, and probably about maybe 10 inches in diameter. Does that sound about right? Yeah.
So it's a small bucket.
Yeah, it's a small bucket. That's right. Okay. That's right. Yeah. The center...
Jonathan, are you guys looking at our live page?
No, they're not.
Okay, I found an image that There's about a minute and a half into your little video.
Yeah.
There's a picture of something that looks like what you might be describing, and I found a picture of this. It's labeled Cyber Physical Systems Expedition Premonition. I found a picture of this from Getty Images that I've just pasted into our show notes.
Yeah, I think that's what he's talking about. Yeah.
Yeah. Okay. So that one you're seeing there was our very first prototype.
Yeah, it looks bigger than what you described.
Yeah, right. That's bigger. So yeah, that's.
It looks a little like the Watergate Hotel.
Right. Yeah. So the new version, the one that we're getting ready to field deploy is quite a bit smaller than that. But that was the original design we used to really prototype the sensors and this kind of door closing mechanism. But as the way prototyping works, you often build something bulkier and heavier in the beginning to validate, and then you optimize. So that's where we are now. So then when we extract the samples, you can from that point you have the data set that comes along with those samples. Our initial experiments show that this data set can be very interesting just beyond training the device to be more selective. It can reveal information about, let's say, mosquito foraging behavior. For instance, when we put it in a greenhouse full of Q-like squinca fasciitis, which is a mosquito that kind of annoys you when the sun goes down, you will see the circadian rhythm of that mosquito in the data set, because you can see when they're encountering the trap, the instant they're encountering the trap, and the rate at which they're doing that. So it will reveal a lot of information, even about mosquito behavior. But then once we get those samples out, you could route them to sort of a standard analysis tests for certain pathogens, maybe PCR-based tests, maybe ELISA-based tests. But what we do is we take them, we extract we mush them up, we extract their DNA and RNA, and we turn them into data. Because again, coming from a tech company, we want to say, how do we turn everything into data?
How do we make this digital? Yeah.
How do we make this digital? And we put them through a gene sequencer. And from the overall sort of view of trying to detect both known and possibly unknown, possibly novel pathogens, the gene sequencer makes a lot of sense because it's a single laboratory step whose cost is decreasing exponentially fast and is broad spectrum. And it's not going to be selective towards a particular pathogen. And then basically I hand that data to Jonathan, who now has to figure out how to make sense of it, because it's a really interesting, complicated data set. So maybe I'll let Jonathan talk a little bit about that side of it.
So you have are you doing the nucleic acid extractions and the sequencing there at Microsoft?
We're not. So we've been collaborating. I should mention that this project, the scope of it is pretty large. And even though we have a really amazing research team here, there are still many parts of this project that are outside of our box, like field entomology. We don't have any field entomologists in this.
So don't feel bad. Nobody else does either. They're not making that one anymore.
So what we've done is to partner with universities like Johns Hopkins, University of Pittsburgh, University of California, Riverside, to build a team that brings this sort of holistic set of expertise. And so in terms of the nucleic acid extraction protocols and the gene sequencing, that's been done with Jim Pippas at University of Pittsburgh.
Okay.
So he's the one who has designed those protocols. They've tested a number of protocols from sort of total DNA and RNA extraction to ribosomal RNA depletion, to some kind of mechanical filtration and amplification of viral particles. But that's something that they have a lot of expertise in. They have been working for many years in novel virus discovery using metagenomics. And Jonathan has been working closely with them to try to better develop better algorithms to make sense of the data that's coming out of these mosquitoes.
So Jonathan, you have one mosquito. What are you actually sequencing? The genome or the transcriptome or both?
Yeah, I mean, that's one of the things we're exploring right now. I mean, right now we're pooling mosquitoes and we're doing RNA-seq on them. That's mostly because our goal is to find RNA viruses, is what a lot of the nasty ones are. So, we might as well start at the RNA. We know from the previous work that we can do things like targeted PCR to do blood meal analysis and to find viruses that we're interested in. So even if all else fails, we know we can do that. But our goal is to really broaden this and, like Ethan said, be unbiased and see what else we can find. And if there's one field that's exploding right now in general, it's metagenomics. So we're trying to leverage a lot of the work there, and start with the processes that have been developed to identify viruses in human samples and see how we can scale them. And really where I think the issue becomes a little more challenging is now our metagenomics is not viruses or microbes coming from a known host, where typically the first process is throw out all the human or whatever the host is. Mm-hmm. But now our world is really all known vertebrates, as well as the mosquito, which, there's 3,600 mosquito species that are hugely divergent from each other. So we don't know exactly what to throw out a priori.
That's a big world out there.
Yeah, so a lot of it is just taking the existing algorithms and trying to scale them and try to make them more robust to this world where we have 50,000 roughly reference genomes that we can try to match to, and they're redundant, and species have similar regions, and they're divergent within species. And of course, many of our references have, shall we say, a lot of incorrect sequences in them.
Yeah.
It would turn out we all evolved to have the same Illumina primers in our genome.
Yeah.
Yeah, right. So you can eliminate a lot of those mosquitoes because they don't feed on people. Although you might be worried about them jumping from animals to people through a bridge vector, as they worried about for West Nile, for instance, but that didn't pan out.
And that's the really exciting parts about this, right? I mean, monitoring the human population is interesting of itself, but of course, many of these viruses have some natural host, some of which we know and we still can't sample, and many of which we don't know what the natural host is.
Right. But this way you get both, right? Because you get the blood meal from the host and you get the virus from the host also.
That's right. I mean, we essentially get the full microbiome of a mosquito, which I include in that statement, the actual microbe community of the mosquito, the microbe community of the host, and the host itself.
Right.
And really the limiting factor there is, we're working with about, 2.5 microliters of host blood.
Right.
And that's being digested. So it depends on how the mosquito and how much it actually ate.
So how many drones have you imagined constructing and turning loose on the world to get this data completed?
That's right. Ethan hasn't told you about the most interesting part about this.
Yeah,
the deployment.
Right. Exactly, yeah. So in terms of scaling, I started on the trap end, which is designed to remove that bottleneck of just collecting the right samples from the environment. And then on the other end of the spectrum is how you turn those samples into data to try to pull out the interesting signals. But the other bottleneck that sits in between those two steps is just getting these traps in and out of the environment. And this is something that, every public health organization in the world that worries about mosquito-borne diseases does probably on a daily basis. They drive out, hike out to places where they're going to put these devices into the field and retrieve them. We did a very early experiment with the island nation of Grenada to do this ourselves, to really understand what it means to do this. They worked with us to try to cover Grenada with traps at roughly every 5 square miles, so that we cover many different habitats from urban, jungle, peri-urban. And what we see really is that this is a major bottleneck. Probably we could deploy on average 6 traps a day, and that's working 12-hour days. Because in a rural environment it's slow because you may park a vehicle and hike out into a place that's interesting to sample. Particularly in the tropics, that can be very slow. And then in an urban environment, even though you might have good road networks, it can be slow because you have a security issue, right? You need to put these devices where they're secure, and typically that isn't the place that's most easy to access.
That's right.
So it's also, in many cases, not the place where the mosquitoes would be.
Exactly. That's also a great point. That mosquitoes do not homogeneously exist throughout the environment. And so you're absolutely right. In the urban environment, there is also the point that you have to put them where the mosquitoes are. And if you take the notorious Aedes aegypti, Aedes albopictus mosquitoes, which are urban, particularly Aedes aegypti, urban container breeders, then their density can be quite localized and can move as it rains, as it dries up, as people add containers to their yards. And so really there's a major bottleneck there that public health organizations have to really go through with their eyes, find these places.
Yeah.
It's pretty incredible, actually.
Plus, you've got day feeders and night feeders to worry about too. So when do you deploy your traps and when do you collect them? That's
and so that's
another layer on top of another layer, right?
Absolutely. So we also want to ask the question: is there technology that can help really give us the throughput we want by deploying and retrieving these traps more efficiently from the environment? So there doesn't need to be a person per doing these deployments. And here there's been a lot of movement in the drone space on using drones to basically fly over trees, fly over buildings, and get into the environment efficiently. Of course, it's not just Microsoft that has recognized that that's the use of drones. It's been recognized all over the place. I think we're unique in viewing this as a problem of making drones very safe and very autonomous. So very safe, meaning, if a drone is going to deliver a mosquito trap around you, that drone had better be running the operating systems and the control algorithms to make sure that it's not going to crash. So you're going to trust it to be doing that job. And it has to be autonomous, and that it has to make decisions on its own. And those can be hard decisions, like, do I place a trap here because that might be a good place for Aedes aegypti, or do I place a trap there because that might be a good place for Culex, or do I avoid this location because I know there's not going to be any mosquitoes there? How do we give it that kind of intelligence that a field entomologist has, or someone who works in a public health organization has,
or that a mosquito has?
Or that a mosquito has, right? Absolutely, yeah. And so this is something that we have been that we're very excited about, and it really leverages a lot of technologies, I think, through the whole computer science stack, really, all the way from operating systems up to machine learning and vision. I mean, the way that we're trying to pull this off is to have a drone that can safely navigate the environment while surveying it. Surveying it means that it's watching the environment with a camera. And from that camera, it's going to reconstruct a model of what it sees. And now the challenge is to take that reconstruction and extract from it the features that are predictive of a mosquito hotspot, or even more specifically, an Aedes aegypti hotspot or a Culex hotspot. And the way we do that is basically training the system, just like we talked about training a trap to distinguish, an interesting bug from a boring bug. You have to train a drone to distinguish an interesting place from a boring place, and you do that by taking video, building these models of the environment, and combining it with data that you've collected, ground truth that says, at these sites you will see a lot of these nasty mosquitoes, and at these sites you won't. And basically you put this into vision and machine learning algorithms. They shake that data around and they try to extract features that are predictive. So the long-term vision then is that gives the drone the autonomy to find an interesting location. If we can get the safety and the autonomy good enough, then we can perhaps even trust the drone to do these kind of deployments in a complex, unstructured environment.
And they would be deploying this entire trap, right?
That's right. Yeah, that's right. Yeah. So, the trap the design that we'll be field testing soon, like Jonathan mentioned, that weighs about 5 pounds. That's unbaited. That's without any CO2 lure. 5 pounds is something that a capable drone today can already lift. Realistically, we would want to make that even lighter. But part of the design has always been, it needs to be light, it needs to be power efficient, because in the end, we do want it to be deployable with an automated system. And if you don't take that into account up front, if you continue to lug around the lead acid car batteries, these traps today,
or the hamster that runs on the treadmill.
Right, the hamster runs on the treadmill. Then
you have to feed it and water it.
Right, and that all has, water is heavy.
Oh yeah, absolutely.
Particularly it's heavy water.
So you would deploy them, and how long would you leave them in the field? Does that depend?
It depends. I would say the usual protocol is on the order of 12 to 14 hours. So because mosquito densities are usually lower than a laboratory setting. I would say usually, because I have experienced some places recently where I do think they're about equal to which is why I'm itching today. So usually you leave them out longer because you don't have that density, and you need the time for the lure to disperse into the environment to attract them. You don't leave it out longer than that because if you leave it out too long, they will die in the trap, and they're digesting the data that we want.
Right.
A couple of questions. Would the drone stay with the trap while it's out?
Yeah, that's I think an interesting question. At the moment, our design is that the two are separate, and the reason for that is really sort of a cost-benefit trade-off. We mentioned the importance of having these traps in as secure of a location as possible, but we also recognize that it's probably impossible. You really can't guarantee site security 100%. So the way you mitigate that is by making the trap as low cost as possible. So it's okay if you lose one.
Okay.
Drones, even though they're low cost compared to aircraft, do you want to leave a high-end drone in an environment for 12 to 14 hours? It might make more sense to decouple the two, have that device, which is inherently more expensive, interact with sort of an insecure environment as little as possible, and then get out of there.
Well, and from an efficiency standpoint, too, you can have the drone deliver 5 or 6 different traps to different places.
Yeah, that's right. That's right. Yeah. So if drones were, arbitrarily inexpensive, but probably there is some, floor to how inexpensive they can become because they still need motors, they still need good lithium-ion batteries. There's some floor to the baseline cost for a drone. I think it probably will continue to make sense to keep them decoupled. But if that floor weren't there, then you could totally imagine a design where the trap utilizes, has a flight mode and a ground mode where they're totally coupled. Yeah.
So I was also wondering whether there might not be a way to preserve the samples once the insect is trapped in the trap.
Yeah.
So you don't have to worry about this things decaying over time.
Yeah, that's a wonderful question, and it's something that that we actually have tried to think about in this design. So part of the advantage of having these small cells is that they can, in principle, create a kind of microenvironment, which is which is decoupled from the environment in which the trap is sitting, which you can't do if you just have a big bag, right? Which is usually a mesh bag. So we did think about, is it possible to include into these small cells a desiccant, let's say, which is released when the door closes, and now you don't have the cell exposed to all that humidity, so perhaps the desiccant would hold up. There's also things like RNA later, which seems to have good results in stabilizing nucleic acids at room temperature. So we actually do have that potential to add that into the design. At this stage, I think we just don't have enough data to make that comparison. So Trap right now doesn't have that in the design, but we do recognize that would be really interesting. It could change the time constants in two ways. One, you don't have to pick it up right away because the samples are stabilized, but also if it can effectively knock down the mosquito and stabilize it at the moment it's caught, then you don't have that 14-hour delay where a blooded mosquito flies in and then it still has that 14 hours to digest your interesting data. Right. But yeah, I think from our collaborators who've worked a lot with the mosquitoes, sort of the conclusion is it makes sense to do an initial baseline comparison. What do the samples look like when you keep them alive? Because that's what everybody else is already doing. They're keeping them alive. Right. And then we can understand, is it possible to optimize, to stabilize in situ? Of course, there are lots of engineering challenges there. If it's a desiccant and you are often sampling mosquitoes at 90 environments with 90% humidity, that really needs to be sealed off well. And for things like RNA later, I don't know how well they've been tested on, so they haven't been tested as far as I know on taking, let's say, a live insect, dropping it into RNA later, and then in a humid, hot environment, and then coming back and seeing the quality.
Another problem that I thought about just now also is that, are you distinguishing between mosquitoes that have fed versus those that have not fed? Or are you just taking any mosquito that is attracted to the trap and taking them back for analysis? Because a lot of these viruses are not vertically transmitted, and a lot of these mosquitoes that just hatched have not had time to actually encounter a host.
That's right, yeah. And that problem is one that's pervasive through all of these trapping systems. You're absolutely right. And it would turn out that these host-seeking traps, which are the ones I'm describing that use a CO2 lure that are attracting the mosquitoes in the host-seeking phase, there's no reason to think that those mosquitoes should be full of blood because
that's right.
Because they're hungry, right?
You bet. Well, they just might have hatched just an hour before.
Absolutely right. In which case you get 5,000 boring mosquitoes.
Yeah, that's right. That's right.
Yeah. But
but you could approach that with a different trap design. I know there are gravid mosquito traps that seek them at the egg-laying stage, for example.
Right, and that can be good for mosquito-borne viruses, where now the mosquito has.
Alan, that's way after the blood meal's digested, though, so you miss out on a whole bunch of stuff. Right. The thing I was driving at, I guess, was that what was your question?
Well, I think the question was about, right, can you find a blooded mosquito? And I just want to say that the host-seeking traps, again, in terms of sort of proven entomological methods, they are proven to actually catch a percentage of mosquitoes that are blooded, and sometimes even blooded in multiple ways. And for the ones that are not visibly blooded, about 15% of those in the trap will have detectable host DNA, even though they're not visibly blooded. I don't know if anybody knows why that's the case, why a blooded mosquito is still going after a host-seeking trap.
Exactly.
But this is, these are the standard kind of traps that are used to catch blooded mosquitoes. Even though you would think, I'm full, why would I fly towards CO2?
So do the wing beat patterns change from non-feeding to feeding? Maybe that's a distinguishing feature you could use in your trap.
Absolutely, yeah. So the answer to that is nobody knows. And we hope...
Good. Yeah, you can have a paper out of that one, I'm sure.
So I have a question, too, though, about have you thought about imaging these guys once you've got them in there? Would that be useful to tell you? I mean, and it goes back a little to a question I had about the training. Initially, it seems like you're going to have entomologists helping you with the training, and if you think that at this point or at some point down the line you're just trapping only Aedes albopictus, but then you get some weird result. If you could go back and look at the image and say, well, that wasn't an Aedes albopictus in that cell, if you had an image.
Yeah. So our protocol for training the traps will involve, keeping images of the samples so that we can, and even probably physically the physical samples themselves, so that we can always go back and look at them. I don't think I don't know how much use it would be to actually have the trap itself take that image. When the insect enters the trap, and the reason I say that is what I learned about speciating mosquitoes morphologically is that it's just super hard. You need to put them under a dissecting microscope, and if this hair is 2/3 longer than that hair, then it's this thing, otherwise it's that thing. So that's something that's going to be really hard to pick up from any kind of low-cost imaging device that you could integrate into one of these traps.
But remember, of the 3,600 species of mosquitoes, they're not all found everywhere. So you've got regions to narrow your search down to a reasonable number, I think.
Yeah, and the differences between genus can be bigger, and whether they're can be different.
Sure. So let's assume that everything works perfect, and your drones are great, and your traps are terrific, and your DNA analysis and all this other RNA analysis is perfect. All the lab tests come back positive, so it's a big go. Where's your field test going to take place? Where are you going to try this first?
Well, that hasn't been totally sorted out yet. We're in discussions with public health organizations in the southern United States, where the mosquito season is already upon us,
so to speak.
Yeah. But right now we're really in having those discussions, because I think, like I said, the basic components are validated to the point that we can really start planning those protocols. But we're still in the planning phase. I do hope to be able to give more details about that in a month or so. But I would just say in a month or so, probably we'll be able to say concretely where these things are sitting and what kind of data they're collecting.
So I was wondering, droids don't have drones don't have a huge range. So if you want to do global coverage, which I assume at some point you'll want to do, you're going to have to be working with local people, right?
Absolutely, yeah. So I think even for a pilot, we need to work with local people who really know what they're doing. They understand the areas. They understand all of it because a system like this is really a combination of of technology, of biology, and it's also a social engineering problem as well. Are you comfortable with a system like this in the environment? And part of making sure that people are comfortable with it is working with these organizations that understand the needs of the local population. They understand sort of what they find acceptable and what they don't, and making sure that that's conducted in a way where everybody feels that it's beneficial. So I think collaborating with local public health organizations is the way to really do it. Absolutely.
So Jim told me that your goal one day is to have real-time computer map where anyone could look and see what mosquitoes are carrying what viruses globally. Is that correct?
Yeah. So if you I always like to use this analogy with these street view maps, right? Now you can go online if you want to know what a building looks like. You type in the address, you get a 3D picture of it. And that was enabled by a similar technology that somebody will drive a car around augmented with a good camera, and they will just take a picture of every place that they've driven. And that provides a data set which sort of unlocks all sorts of interesting capabilities that you didn't have before. Those systems scale by initially providing small data sets about, let's say, one city. And that data set is interesting enough that it allows the system to scale to multiple cities and then eventually globally. So that's sort of the way that we view this. If, let's say, in cities that are hotspots that really need to address mosquito surveillance in new ways, and of course, in the context of Zika, that's something that we see happening that's very pressing now to increase the throughput of mosquito surveillance. Then you have kind of a bootstrapping process, right? Where you try the system in a place that makes sense now, you get value out of that system, and then you allow it to scale. And the more it scales, the more you go from local information to global information. I think what Jonathan was alluding to in terms of the metagenomics is you could just imagine as you go from these smaller scale experiments to larger scale experiments, not only is the metagenomic data huge, but the inferences you want to draw on top of that data depend on pulling out high level signals with some confidence. Right. So a virologist can't go back and look at each of these metagenomic data sets. You will just have too many of them. Right. And the challenge will be trusting that the algorithms can really pull out high level interesting signals and tell you with some confidence that those signals are really there, so that you can do exactly that. You can scale from local places to global places while being sure that the inferences are somehow actually useful.
What's your time frame for global deployment? Are we talking about 5 or 10 years, realistically?
Yeah, I mean, I'm always of the view that if the technology trends are clear, that it just will happen. Yeah. And I think the technology trends here are so clear. The three trends are, making field entomology higher throughput by using modern technology, low-cost microprocessors, using exponentially decreasing costs in metagenomics to give one lab step to turn biology into data, and then of course there's the scale of cloud computing, which allows these algorithms to scale. So when you put these three trends together, I think it's just totally clear that the convergence will happen one way or another. So I will just randomly say 5 years. It could be half that. You never know with exponential pace of technology, but I think it will happen. Yeah.
So I was at a TED Talk. Actually, I was giving a TED Talk, and I was waiting for my turn. And your illustrious leader, Bill Gates Jr., was on the stage with Chris Anderson.
Did he release mosquitoes?
Yeah, he did. He did, actually. So it's kind of an irony and a complete turn of circle that here Microsoft, instead of releasing mosquitoes, now wants to trap mosquitoes. So I think that's a great progress that's shown even within your corporation. So give Mr. Gates my best when you say
But it's certainly good progress that they're going to be removing bugs from the world now. Yes,
yeah, exactly. That's the other point I...
Sounds like we have to get both of you back if you're still there in 5 years.
Oh, we want to see this thing in action.
Absolutely.
See how it's gone.
Yeah.
Sounds good. That's a deal. Are
you collaborating at all with Tony James, by the way?
No, I don't know Tony James.
Okay,
he's at UC Irvine. Yeah.
UC Irvine. He's the first one to ever transfect a mosquito successfully. This was some time back, but I know he's intimately involved in this kind of research. I would highly recommend just calling him and saying hello.
Yeah, and I'm sure our colleagues at UC Riverside must know his group.
Oh, they do. Of course they do.
Yeah. Cool. Well, I have to apologize. Like I said, we're sitting in winter.
Sir, Mr. Gates is at the gate.
I got it.
Yes, and
well, thank you both, Ethan Jackson, Jonathan Carlson from Microsoft Research. We really appreciate your time.
Thank you all. This has been a lot of fun.
It sounds like a great project, and boy, good luck.
Thank you. Have a great day and sunny
wherever we are.
Well, remember, somewhere in the world it's always raining.
Yes.
Yeah, somewhere else. Yeah.
Bye.
Okay, thanks. Bye.
Bye.
Yeah, what a cool project. I want to work on that.
That's
I don't know what I do though. I don't have anything to contribute. I could do PCR, but they don't need me to do that.
They don't need that. They want to automate that.
They're going to automate everything. It's so cool, and then the data they're going to get. Wow.
Right. Hope you're right.
This is great. Hey, Rich Condit, do you have some offspring at Microsoft or something like that?
I did have offspring at Microsoft. So Jeremy worked at Microsoft Research from 2007 to 2010. Then moved in 2010. He moved to Google. I was trying to, I nearly mentioned Google several times during this, but I kind of kept it quiet.
You didn't mention that the mapping was Google.
Yes, exactly.
Right.
So there's one guy on this team, Shaz Kadir. If you look at the profiles of these guys, who Jeremy actually has published a paper with.
Nice.
So he worked with Shaz when he was there. I'll put a link to the paper in the show notes so that you can see it. Of course, I can't understand even the title, but and I corresponded with Jeremy. He says he doesn't personally know Ethan Jackson. I didn't mention Jonathan, but I'm sure that if Jonathan or Ethan knew Jeremy, they would have perked up at the last name. Yeah. But at any rate, he worked with Shaz.
Cool. Nice. Wow, nice stuff. Really nice stuff.
Yeah.
Nice to see how broadly based they are, too.
Yeah, it's great.
The other thought I had was that if these drones are big enough and robust enough to carry these traps, maybe Amazon can partner with them and have packages delivered at the same time.
They've been talking about that, yes.
I'm sure.
You hear, Dickson, that Amazon is making an autonomous car.
Yeah, right.
And if you get one and deliver packages for them, you'll get some money back.
You get some money. Right.
This episode is sponsored by CuriosityStream, world's first ad-free nonfiction streaming service with over 1,400 titles and 600 hours of content. It was founded by John Hendricks of Discovery Communications, so you're guaranteed access to real science shows, not reality TV currently plaguing cable TV. You can watch it on many platforms on the web: Roku, Android, iOS, Chromecast, Amazon Fire, Kindle, Apple TV. You can get it in 196 countries. And they have, of interest to our listeners, a wide variety of science and technology content and nature, history, and many more topics. They have, for example, Stephen Hawking's universe. They have a number of programs on viruses, one called Destruction and Creation, an original where they talk about Zika virus, Life on Us, a series that explores the biodiversity of our bodies, Jason Silva, Transhumanism, Secret Life Underground, and many, many more. Monthly and annual plans available starting at just $2.99 a month, less than a cup of coffee or the cost of an online title on the competing on-demand platforms. Enrich your life and continue learning with one of the largest 4K libraries on the internet. They have over 50 hours of 4K. Not that I could tell the difference between 4K and 1080, but check out curiositystream.com/microbe. Use the promo code microbe during sign up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe. Use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiV.
Yes.
Oh, dear. I just lost my thing here. Okay, here we go. Rich Condit has a follow-up about mosquitoes and viruses. Did we say something that bothered you?
Listen, I'm not bothered. Okay?
He's retired.
You're going to have a hard time bothering.
Can I have some of those genes?
Pills.
It helps to retire. Okay, so It has been stated numerous times on TWiV that mosquitoes that transmit viruses only do so when the viruses replicate in the mosquito. And I had this little thing in my head from McFadden. I had heard him say many times that myxoma virus, the pox virus that infects rabbits, was transmitted by mosquitoes but did not replicate in the mosquitoes. And so I followed that up. First with McFadden, who said that the work was done by Frank Fenner in the 50s. And so I actually did some research into that and found a couple of papers here that I stuck links into. That's Frank's work on transmission of myxoma virus. And bottom line is that, yes, it's vectored by mosquitoes, and they can find no evidence that myxoma actually replicates in the mosquitoes. And so their conclusion was that it's mechanical transmission.
Wow.
By contaminated mouth parts.
By contaminated mouth parts.
I love it. Contaminated mouth parts.
Sure.
So you don't need a lot of virus, presumably, then, because you're not going to have a 10 to the 3, 10 to the 4. On
no,
the
myxoma, the LD50 for myxoma is or the infectious dose 50 for myxoma is like 0.5. I mean, if you get one myxoma virus particle in you, you're dead.
Wow.
If you're a rabbit, that is.
So, Rich, are there any virus?
If you're a rabbit who's not adapted to it.
Right.
Not only that, but the myxoma as a pox virus is quite stable.
Yeah.
And there's part of these studies has to do with the effectiveness of transmission as a function of where on the animal the mosquito picks up its meal. And so they show that when a mosquito has actually landed on a lesion that is probably loaded with virus, it is more likely to transmit that to a host. So it's all a matter of numbers. But in this case, the numbers favor mechanical transmission.
Do you know any insects that are infected with a pox virus?
Not personally.
There's lots of insect pox. There are insect-specific pox viruses, yes.
Or vector-borne pox virus infection?
There
Besides this
one. Wow.
Smallpox,
that one I'd have
would that have been
think about
possible?
that.
Like this one?
No, I don't think so.
Like if a mosquito got around the edge of a pustule and then moved to another host?
I suppose it's possible, but it's not been documented as a primary thing. Now I'm trying to think.
Or body lice?
Now, the thing that comes to mind is African swine fever, but that's not really a pox virus, but that's tick-borne.
Right.
And it has a cycle in ticks. I'd have to add, this is a good question, Dickson. I'd have to my knowledge, I don't know of any insect-borne pox viruses where there's actually replication in the vector.
Because, I thought about the early epidemics of smallpox throughout Europe. And, this was a poverty-ridden area. All the metropolitan regions of Europe in those days were hellholes of loss of sanitation. Body lice, head lice, those things are being exchanged around. You sleep in the same bed, and the next thing you know, everybody's got everybody's arthropods chewing on them. So if one of them caught smallpox, I know that it's easy to transmit on fomites as well, but just curious.
So the viruses we've been talking about, the flavies and the toga viruses, presumably they
do replicate.
They do replicate for sure, and maybe it's not enough what's left on the mouth parts, contaminated mouth parts, because they need more to infect the host. I would
Something
guess.
Like that, yeah.
But it just goes to show in biology, there's always an exception to whatever rule you state, right?
Question.
You should always be careful.
No question.
Yes.
Well, all the weird stuff happens in Australia anyway, so.
Yeah, we talked about that last time, didn't we?
Yeah.
It's come up a few times.
Carp, getting rid of the excess carp.
That's
Did
right.
you hear about that, Rich?
That's right.
Yeah.
Yes, I did. I listened to TWiV.
You do?
do.
When you're not on, you listen to it?
Even when I'm not on, yes.
That's great. I appreciate that. We all appreciate it. We have a bunch of follow-up of various sorts, mostly dealing with preprint servers. There's an email from Chris who sent a link to a paper which shows videos of mosquitoes probing for blood vessels.
Yeah.
Which we talked about last time. It's pretty cool.
Yeah. If
Very
you
cool.
watch, The probing can last for more than 6 minutes.
6 minutes, yes.
Yeah.
That
How
would
do you
be
know you don't have a mosquito on your?
well, an
that
animal,
long?
an animal,
Oh,
right?
An animal, yeah.
Well, or you when you're asleep.
Right. Yeah. No,
Yeah.
that's true.
So if you look at their experimental setup, it's one of these things where they've got an anesthetized mouse who's had its skin sort of splayed out over a microscope stage, and so they have living tissue that they're exposing to mosquitoes, and they're taking microscopic pictures. What I really like is there's all this probing that you see going on at the microscopic level, and then when the proboscis hits a capillary, it just freezes.
Exactly.
It just instantaneously freezes. It knows
It's a
when
heat-seeking
it's
missile.
struck gold.
It is a heat-seeking missile. Quite literally.
All right. Then we have a bunch of emails dealing with preprint servers, which people are clearly interested in. Steen wrote over. So he wrote a comment at Virology blog where I first wrote about the bioarchive articles. As discussed in great length on TWiV 388, the paper described here has not yet passed formal peer review. I, for one, could not care less. I know that the author of this blog peer-reviewed the paper, which provides more confidence than knowing that 2 or 3 unspecified anonymous scientists reviewed a published paper. I would rather read a paper without the cognitive bias introduced by knowing that it was accepted by a prestigious journal. If serious controversy arises around a preprint, that might result in more stringent formal peer review of that paper, which will probably be a good thing. Someday a preprint will attract public attention and will later definitively be shown to be wrong. This may damage the public trust of researchers and of science communicators. In the meantime, slow dissemination of results and uncritical acceptance of formally peer-reviewed papers are much bigger problems. Thank you for publicizing preprints. I hope you revisit this paper on TWiV once it inevitably gets published somewhere. Thank you, Steen, for your confidence. I really appreciate it.
Can I ask for a point of clarification here about preprint servers? Because I'm an old guy, right? So
some of us are.
Back from the time
I got you beat, Rich.
So am I, and so is Dickson.
Very old.
When preprints were, paper.
Mm-hmm.
So I grew up thinking that a preprint was something that you prepared a paper for publication, and then at about the time you sent it into publication, some people would wait till it was accepted, but in my tradition it was when you sent it in for publication, you took a copy of the manuscript that you had submitted for publication and distributed it to people that you thought were interested.
Yeah, that's right.
Okay?
That's right.
So, preprint servers.
You just put them online now. That's it.
Yeah, but is there any assumption that the paper that's being put on the server has actually been submitted for publication somewhere?
No, it doesn't have to be.
No.
You could put something there that you never submit. You just want to share it.
Fine.
Yeah.
Okay. I just wanted
and
that
this is based on a tradition from many branches of physics and math, where publication traditionally involved sending copies of a letter around to your colleagues.
Mm-hmm.
And so, somebody would send out their physics paper and just distribute it informally among people who they thought would be interested, well before they prepared it for publication.
Actually, we just got off from talking to 2 guys that pursue a different model. In computer sciences, they're all conference papers.
Right.
Mm-hmm.
Right? So that's a whole different model. At any rate, I just wanted that clarification.
Yeah, that's a nice.
By the way, just a quick step back to the pox viruses. I was Googling this, and I did find one paper on mechanical transmission of Capripox virus in goats, as well as African swine fever virus.
Okay.
All right.
And that's a fly.
So submitting your paper to your friends for pre-peer review
There are no friends in
really actually gives you peer review unofficially because they'll send back comments. So that's a good thing, I think.
Fine. Well, you
Very
get
good.
comments on these peer.
Very good thing.
Alan, can you take the next one, please?
Yes. So we're
Todd.
We're at Todd. Okay. Greetings, Twivers. As I was listening to episode 388, What Could Possibly Go Wrong, and the discussion around preprint preprint publications, as I like to think, science-savvy layperson, I would have to agree with Kathy. If you're blogging about a paper that has not yet been peer-reviewed, it is worthwhile pointing that out rather than solely relying on a link to the study. That, at the very least, will let the reader know that they should be looking at the paper through a somewhat more critical eye. I also have to disagree a little bit with Vincent regarding poor quality science published through preprint servers. Vincent mentioned that we would not see an increase in bad papers because scientists want to put their best work forward. While that may apply to the majority of researchers, there are people who are just bad scientists, or even hucksters trying to game the system, that would not otherwise get their papers into a decent peer-reviewed journal. This allows them to get their work indexed and available for citation without worrying about their paper being withdrawn or retracted had it somehow worked its way past peer review. A layperson may not know what caveats to apply to these preprint publications, and they may give a veneer of credibility to crank ideas. Granted, I'm not too familiar with preprint servers and how submissions work, so these concerns may be moot. Perhaps you could comment on that aspect. And Todd runs Harpocrates Speaks.
I'm not doing that. I'm just putting a link because, as Steen said, I peer-reviewed it, and that's good enough for me. and if people come, they come to my site to read what I have to say. They either trust me or they don't. And that's the point. I didn't make it very eloquently last time. I felt I peer-reviewed it, and for me, it was good enough. I don't think we're going to get a lot of hucksters. We got our hucksters in science, but I think the majority of these papers will be from well-intentioned people. But yes, you're right. There will always be hucksters.
Well, and the hucksters have plenty of journals that they can choose from now, thanks to the predatory open access journals that.
That's right.
Right.
You can publish anything in a quote unquote peer reviewed journal.
Right.
So.
Dixon, can you read an email?
I will do my very
next
best.
One, please.
It says, Anthony writes, wasn't Wakefield's research published under peer review?
Yes.
In that very narrow crack that I'm qualified to look through, I see markedly. Markedly. From here it says markedly. That's okay. I see malarkey published in named journals. There's also the same author publishing very similar work in a number of different journals. Something that I find really rotten are the cities.
Sites.
I can't read these emails that when looked up don't support the statement. And that's for facts that are just not applicable. There's also the sites that when looked up result in sites. Tracking them down results in an anecdote. How are these papers reviewed if the sites were not checked?
Well, very few people check the citations.
Right.
That's the bottom line.
I agree.
This is one of the things you routinely encounter in science is you go to the paper and somebody says something interesting and cites a paper and you look up that paper
it doesn't exist.
Say, wait a second.
That's right.
That's not what that says. Or you go through some long chain of citations and you get to something that's just wrong. Yeah. But everybody cites everybody else.
Irrelevant.
Hey, Rich,
Yes.
Rich, can you take the next one, please?
Lee writes, Dear TWiumvirate, I'm listening right now to your discussion of Vincent's use of a preprint in a blog post, and I have to step up and agree with him. I think Kathy's right that a disclaimer would be ideal, but I also think Vincent is right that it would make the blog post unwieldy. I've had to choose concision over total detail a few times in blog posts, and I reconcile it by saying that when someone asks, I can commend their attention to detail and publicly explain why their caveat didn't make the final cut. The real problem I see here is that the public has been led to believe that scientific results, peer-reviewed or not, are settled fact just because someone respected talked about them. Vincent is a peer reviewer or editor for several different publications and was elected president of ASV by his peers. What's the difference between his reading the paper for the purposes of peer review and reading the paper for the purposes of writing a blog post about it? The main difference I see is that with the blog post, Vincent risks small damage to his reputation if the paper is ultimately false. If he were asked to review it, he would read it and analyze the experiments and then decide whether or not the designs and outcomes made the results sufficient evidence to support the conclusions. Isn't that exactly what he did for the purposes of his blog post, but out in public rather than anonymously? Certainly a full review would come with viewpoints from additional scientists, but we cannot just take Vincent's blog post and say, okay, that's reviewer numbers, but can we not just take Vincent's blog post and say, okay, that's reviewer number one's opinion.
Yeah, right.
Sure, he could be wrong, but that's true of peer review too. I think this is what peer review is going to look like in the age of preprint archives, and we better get used to it. Would there be a difference had the paper been peer reviewed and then written about? Vincent would just be reviewer number 4 or 5 or 6,000. Peer review misses falsehoods, both intentional and unintentional. Plenty of peer-reviewed papers are discovered to be wrong by the community, but the veneer of peer review has long given the public confidence in these results, the same confidence that they would have in a blog post written by a prominent scientist. Andrew Wakefield's paper was peer-reviewed, and that gave it legitimacy that it did not deserve. Maybe the problem here isn't that there's a hazard in believing Vincent's blog post. Maybe the problem is that we haven't educated the public enough for them to be widely aware that the same hazard exists in believing any scientific result in isolation, peer-reviewed or not. Lee Feynman, PhD, a.k.a. John Schuyler.
Right, he just went through graduation. I have a comment. Vincent, in his next blog post about a science paper, changed how he indicates the paper, and he puts in parentheses, link to paper. And it's then a different color ink for the font, and that's clearly a hyperlink. All I was asking was that same sort of thing be done for a preprint. So parentheses, link to preprint. I don't find that being unwieldy at all. It's no more unwieldy than having references in a science paper where your eye either focuses on that or can pass over that.
Well, I did that because of your comments 2 weeks ago, because I normally don't do link to paper. I just, embed the URL in the name of the paper or something.
Right, which was my objection in the first place.
But I don't think that, so the problem is that you can't explain that this is a preprint and what the ramifications are.
Just put a link to preprint.
Yeah, but is that what you said originally?
Yes. Just put link to preprint if it's a preprint.
Well, I link to paper or preprint. Do I have to say preprint even?
Yes.
Because what do people know if it's a preprint or not? That's the other part of this problem.
When they link to a preprint on bioRxiv server up at the top, then they will potentially look and see that, it's a preprint, and then there's a link from there telling you what does this mean. That's all I'm saying. It's link to paper versus link to preprint. That's not unwieldy in my opinion.
No, that's not unwieldy
No,
at all.
that's not what we're saying is unwieldy. What is unwieldy is to say this is a preprint, it's not peer-reviewed, and it means this, that, and the other, and you have to go on and explain it.
I never said that you had to say that.
Okay, I think some of the letter writers are thinking that you meant to insert a disclaimer.
Yeah,
well, a disclaimer is to say link to preprint. Just using the word preprint itself is a disclaimer.
Well, I think they heard the same discussion that we had, so either they're misinterpreting it or something else. But
sometimes when people write about papers, they'll say such and such a finding published in Science or published somewhere. Okay, and they'll name the source, perhaps with an embedded link. Does that come under the heading of unwieldy? I mean, Alan, you do this all the time. Do you often in the text cite the sources?
Yeah, that's what I did. So I had in a URL, it turns up a different color on the web page, and you click on it and you go to the source of the publication. And I just did the same thing with the bioRxiv without saying it was a preprint.
A very, very standard thing to do in any kind of online writing is to just highlight a relevant word. As the new research demonstrates, and then highlight the new research word or words, and that will be your link.
Mm-hmm.
Yeah, so I just added, and from this day forward, I shall, for whether it's peer-reviewed or not, I will put it in parentheses, link to paper or link to preprint. Okay.
Which is also, that is also widely used. I don't think it's as widely used, especially in blogging. People are more likely, certainly I am more likely to just link to a word, but I'd never really I'd never come across this problem of linking to a preprint versus linking to a paper.
Do you see any advantage in naming the journal or whatever or preprint server where it was published? Some people do that. As published in Science, as published in Nature,
right.
blah, blah, blah.
Yes.
Published in BioArc.
You could do that. You could say that too. That's fine. But I don't want to do that. I'm going to just do it as I've said. But
I can see the argument for all of those. The nice thing about just linking to a word in the sentence is that you've got just the sentence, and if people don't feel like following the link, they don't. It's not an interruption.
Right. It doesn't interrupt the flow. Yeah. Right.
But I think that Kathy's suggestion of parenthetically a link to paper or a link to preprint is a pretty good compromise, in that using the term preprint should tell people what they're looking at. And if they're not aware of what a preprint is, they can Google it.
Mm-hmm.
Can you take the next one, Kathy?
Is that
Mike?
Mike. Vincent et al. I was interested in the dialogue among the group regarding the use of preprint servers to publish work prior to peer review. While I appreciate the desire for scientists to get their data published, I wonder about potential conflicts when submitting a manuscript of previously published data to a peer-reviewed journal. As you know, many journals require that authors indicate where the submitted data has previously been presented, for example, meetings, or that they document that the data has not been previously submitted for publication or published elsewhere. It seems to me that submitting a paper to a preprint server prior to submitting to a peer-reviewed journal could be viewed as double dipping, particularly if both publications are cited in a CV, or if claims for publication date priority are put forth. We all know that meeting abstracts are not treated as proper peer-reviewed publications, hence the separate section in one's CV, and are not particularly relevant when questions of who published when arise. I certainly see the value in preprint servers for reporting of studies, but I also see the potential for considerable abuse by those who are trying to pad their CV. I guess institutions will need to add an additional section to their CV template for non-peer-reviewed articles, and journals will need to find a way to screen preprint servers to ensure that submitted studies are novel and not previously published, if that is a criterion for submission. Note that these are my opinions and do not necessarily represent those of the federal government or Fetal. Regards, Mike. Well, it turns out that for the template that the University of Michigan Medical School suggests for CVs, they have you put non-peer-reviewed articles and book chapters in a separate section. So they have you separate that out. That's not unusual.
Yeah, and I think very, if it hasn't already happened at institutions that didn't already account for that, they certainly will very soon.
This issue of journals, some journals will not accept a paper that has been put on a archive, a peer preprint archive. Some will and some won't, right? So that's quite clearly stated in the journal's webpage, so that's not an issue really.
And actually, a lot of the major journals have come out and said, especially recently with all the stuff from Zika going on the preprint servers, have come out and said that they will accept papers that previously had previous versions on a preprint server. So they're shifting a little bit on that, but
some do not.
It'd be nice to know if somebody has a compiled list of which ones won't and which ones will.
And the other thing is, I think a lot of people are still distrustful of whether the journals will actually abide by that. Because a research editor at a journal can just reject your paper out of hand and not really.
Yep.
They could just say it's not novel enough.
All the ASM journals will take papers that have been put on preprint servers. They embrace it fully. You get a publication date on a preprint server that is acknowledged. I don't know what value that is, but you can say I published this on a preprint server on this date. But the value of that going forward, it remains to be seen.
Right.
Did anyone not read? No, I think everyone has had a round. We just have a couple more. Two more. John writes a few comments about preprint discussion. The comparison of conference presentations and preprints reminded me of this Retraction Watch post about thousands of retracted meeting abstracts. And there's a link to an article. One publisher appears to have retracted thousands of meeting abstracts. Yes, thousands. The previous TWiV, Quaxed, gave 2 reasons why preprints in biology are more of a danger than preprints in physics. First, a paper about a disease or a cure can be worth a lot of money. Lawyers can get rich, drug companies can get rich, investors can get rich, even if the paper is later discredited just by betting on changes in stock price. That is why journals started requiring competing interest declarations. Second, publishing bad statistics can cause real-world consequences. You discussed the subgroup of MMR vaccine recipients that showed a higher risk of autism. To me, it sounded like an example of the old saying, 1 in 20 2 sigma results is wrong. There are people who take those reports seriously. When I write online, I occasionally test my readers by saying something misleading, linking to my source and asking them to spot how I misrepresented it. I'm hoping to teach skepticism. These experiments have been failures. Some of the TWiV panel said blog readers are unlikely to click links and less likely than that to understand the page they landed on. This is why you should distinguish preprints from peer-reviewed papers rather than hoping readers will click the link, see the word preprint, and understand how a preprint differs from pre-publication copy of the peer-reviewed final draft. All right. One more, Alan.
Sure. Deborah writes, Dear Vincent, I found your podcast somewhere around 2010. I'm very nervous in writing to you, but am more grateful than you can know for your efforts to bring science and virology to someone like me. 55, few classes past high school, factory worker, mom. I'm just a little concerned, though. Since I've been listening for a while, I'm used to the light, intelligent, and respectful way you handle all the discussions brought to the table. So when the issue of whether it would have been better had you notated preprint in your blog commentary on the dengue virus antibodies, you seem defensive. You're a wonderful host and facilitator, probably a great teacher, too, but you have a following now, and people Google taking things out of context, believing everything they read as gospel. Not your fault, who knows? You and the TWiV team might be virology's answer to Neil deGrasse Tyson or possibly Carl S. To the team, I love you all. I'm grateful you all attend these podcasts with respect and enthusiasm, but even an uneducated science buff like me knows that preprint means the piece hasn't been printed, hence the pre-prefix. Kind regards, Deb. P.S. Intermittent rain and clouds all day, 40 Fahrenheit, 4C. Ugh. So I think what we can say with certainty is whatever approach you take to citing a preprint in a blog or publication post, it will be considered wrong by a significant portion of the population. So you can, with confidence, just choose the one that you think makes sense.
Exactly.
Yeah, pre is we know that a lot of people know pre, but again, as we discussed earlier, the ramifications are not clear to everybody what it means. Are the data right? Are they not right? So the details, the devil is in the details. That's what we were talking about. And right. I'm not sure that it's clear unless we go into it. I mean, that's what we discussed, that dengue paper, right? We can do that. And that's why I blogged about it and so forth.
And as far as Vincent's sounding defensive, I mean, that's the way he is when he gets enthusiastic about something.
No, I was very defensive because I felt I did the right thing on the blog, right. And I felt that I peer-reviewed it, and a number of people didn't agree with that. So it's not great. As Rich Condit said earlier, nothing bothers him, and I wish nothing bothered me in a way, because lots of things bother me, and I don't like that, but it's my parents. I'll blame my parents.
I used to get intense about things
right.
In the laboratory. The people in the laboratory would refer to me as focused. That became the euphemism for.
Did they try to hand you a Snickers bar?
The best part about this is that Deb is 55, a few classes past high school,
yes.
Factory worker, mom, and listening to TWiV. I love it.
Yeah, absolutely.
Thank you.
Good.
I mean, I'm still annoyed when I see the sign pre-owned vehicles. I'm absolutely pissed off at that sign because that's a misuse of the English language, and that's where this confusion started. There's an
blame the used car salesman.
I'm damn right I will.
All right, I wanted to catch everyone up on Zika. There's so many things being published, and I just wanted to touch on a couple that had to do with mouse models of microcephaly. There are 3 papers and this is the last part of, if you remember a couple of weeks ago, we talked about, what's the name of those 7 different points for proving teratogenicity of something?
The Shepherd's criteria.
So the one missing was having an animal model
Right.
And the other one was the Bradford Hill criteria.
Yeah, and this is it now. These 3 papers show effects on fetal brain. First one is in stem cell.
And this is open access.
Open access. It's called Zika virus disrupts neural progenitor development and leads to microcephaly in mice. This is from the Chinese Academy of Sciences. And here they have they have used, which strain? Zika. Which Zika strain is this? Brazilian strain? Yeah, it's an Asian Zika strain isolated from Samoa, a patient in Samoa. So they injected they have pregnant mice. And they inject right into the brain of the embryos.
That's tricky.
It must be tricky.
Yeah, I really wondered about this. As a matter of fact, exchanged some emails with Kathy about this. They must take the pregnant mice and
Do surgery
to inject the
You think?
embryos. Yeah, I mean,
You're going to see where you're putting the needle.
And then
you can do some image.
sew them back up, and these things, the mice actually deliver these pups.
Well, then they eat them all.
on.
Or they eat the ones that are affected, but they don't tell you that in the methods.
No, they don't. They don't say you're doing surgery. That's why I asked Kathy if she knew anything about
Couldn't
this.
you do some fancy imaging to know where to put? Why not? Why not?
Because there will be multiple pups inside, and you wouldn't be able to tell where the needle's going.
But you could stereotactically put a needle in a very small part of the brain.
Well, then they really should have described how they did it. But
Anyway, they inject which is really interesting. As we'll see in the subsequent papers, you don't actually have to do this.
Right.
And I looked up the phylogeny of this isolate, and it is very close to the stuff that's circulating in Brazil now. And all this phylogeny becomes important.
Yeah. Anyway, the virus replicates in the injected animals. The virus replicates in neuronal progenitor cells. And basically, they have a nice picture of the brain from infected versus control, and it makes the brain slightly smaller. It destroys the progenitor cells.
Given enough time, it pretty much replicates in everything, including post-mitotic neurons.
Yeah. Of course, if you're replicating the progenitors, that's bad because then you don't have many neurons, and that presumably makes the brain smaller.
Right.
And they say it not only replicates in these progenitors, it suppresses their proliferation. They have a normal proliferation program where they would differentiate into neurons. It interrupts that. And also, they looked at gene expression in these animals, and there are a number of genes that have been associated, mutations in them have been associated with microcephaly in humans, and these are downregulated in these infected mice. Now, interestingly, the infected mice are eaten quite quickly after being born, so they can't study them, which is really... but that's something that would happen, because the mothers know that the pups are not right, so they eat them. But so apparently, this virus causes what they call microcephaly, although, as Rich pointed out, it would have been nice if they had put in an older African strain, which we think is not associated with such spread in humans, and would be nice to know if that did not cause microcephaly, or if it did. I don't know why they didn't do that, right?
Well, we'll see it.
No, we don't.
In another paper.
But they don't actually put it in mice in the same way.
Right. Okay. So they didn't put it in embryos, but they're probably working on it. But we
maybe
see a similar thing with one of the other papers. Is all I was saying.
Maybe it didn't. Maybe it didn't work. Maybe they've all done it and it didn't work.
Well, the next paper that we're getting to deals with this a little more.
Yeah, but none of them actually do the experiment of infecting mice with the two strains and see what happens. There are all kinds of peripheral stuff, but they never do the one. I don't know why. The second paper is a Nature paper. The Brazilian Zika virus strain causes birth defects in experimental models.
Also open access.
Also open access out of the University of out of São Paulo and UCSD and a variety of other places. But here they take a Brazilian isolate of Zika virus from the northeast of Brazil, isolated in 2015, and they infect. I believe this is intravenous infection.
Yes, it is.
Tail vein. SJL, two different strains of mice, SJL or C57 black pregnant mice, and then they evaluate the newborns right after they're born. And they find in the SJL mice, they find that they find by PCR virus in several tissues, especially in the brain. And they find a variety of malformations in the surviving animals, cortical malformations, reduced cell number, and cortical layer thickness, all associated with microcephaly in humans, and also ocular abnormalities. Remember, an early paper we talked about a long time ago saw that in people. But in the C57 black, no major issues with those pups.
So clearly, the mouse strain that you use for these is critical.
Right.
Yes.
And we see more of that later on.
Right, and just for people who might not think that much about mouse genetics, you might make comparisons to, different breeds of dogs or different breeds of cats or things like that. So, they're still dogs, they're still cats, they're still mice, but you have a certain strain of them and they have a susceptibility to this virus and another strain does not. Right. And so that it's also important because this is really the first mouse model of an immunocompetent mouse, if you consider SJLs immunocompetent, which in the scheme of things, they pretty much are.
So they have a nice picture of these pups, these SJLs, mock infected and Zika infected. You can see the one mock infected are a certain size and the Zika are smaller. Overall growth reduction, not just in the brain. It's quite nice. Then they do a series of experiments with, they generate human cortical neuronal precursors and neurons from stem cells, and they infect these with a Brazilian and an African strain. Now here they use both, and they previously didn't do that, and that's why I think they should have done it previously, but they haven't. And both of these viruses infect these human cortical MPCs and neurons in culture. And they say there's no difference in neuronal cell death at two different multiplicities of infections. And they do some experiments with three-dimensional cell culture systems, brain organoids or cerebral organoids, again with these two strains, the Brazilian and the African Zika virus strain. And the Brazilian strain replicates well in these organoids, it destroys them, and they say that the effects are less with the African strain. They have less of an effect. So there's a difference between the in vitro differentiated cells and these organoids in terms of the two strains.
The African strains do have some effect, though.
They do. They do have an effect, but not as much as the Brazilian. Then they do a neat experiment with chimpanzee cerebral organoids. And there, the African strain does better than the Brazilian strain, which... Yeah,
basically they say the Zika virus Brazilian strain doesn't replicate.
Yeah, so I guess, the African, of course, is thought to have come from monkeys to humans via mosquitoes, and then the Asian and Brazilian strains have been passing in humans to humans for 60 years or so, so maybe something has happened during that passage, and we're seeing some evidence of this here.
Right, but based on these systems, I wouldn't put a whole lot of stock in the strain differences of the viruses. The strain differences of the mice are definitely
for sure.
Though.
All right, and they conclude by saying, our findings support the hypothesis that microcephaly is a distinctive feature of recent Zika virus Asian lineage virus. But I don't agree with that, as Alan says.
Right.
In mice, anyway, they haven't done the experiment.
I want to see them do the experiment. I think that's an important experiment to do. And they had the virus, they got the system all set up. It's hard,
Yeah,
they should have done the experiment.
It's kind of a tease. It's they dance around it, and we don't get the actual experiment.
One thing I wanted to point out on the previous paper, and then kind of reiterate it for this paper, is that the mouse model in the previous paper had some really nice parallels with what had been seen with the in vitro neural progenitor cell results, and that in some ways helps to validate that in vitro system. And in this particular one, they point out a number of ways that the phenotype of the infection in the mice recapitulates the phenotype of the infection in people. Again, sort of cross-validating these models. Now, there's things that we know that are going to be different in mice from the human, but there's no perfect model. But I think so far these are validating some of the other they're validating the in vitro model, and the similarity to human helps validate these mouse models.
And on a side note, if you look up at the top of this paper online, you see that it is a near final version.
Yeah, I saw that.
So this is when Nature does advanced online publication. Usually what goes online is the final version of the paper. They sometimes, if they think it's a really time-critical issue, like Zika virus, they may put what they call a near final version. So this may be revised before the final version gets printed.
Yeah, there are a number of typos in all of these.
Now, I don't know. I don't know if the step from near final to final would include something like an additional experiment.
Yeah, I don't know.
But
well, we'll see when it comes out. Yeah.
Just
anyway,
revising your
germane to the
conclusions.
preprint discussion.
Yeah. The third paper is from Michael Diamond's lab at WashU, Washington University, and it's called Zika virus infection during pregnancy in mice causes placental damage and fetal demise. Yes, of course. This paper uses a different mouse model in which the mice either lack the gene for the type 1 interferon receptor, or they give the mice antibodies to the type 1 interferon receptor as a way of suppressing the innate immune response. And this model was published previously by Diamond, but not with pregnant mice. In this study, they're looking at pregnant mice, and they are
I believe the previous publication, Lazier was.
Yeah, she was on it. That's right.
Mm-hmm.
Right.
This one uses a French Polynesian Zika virus isolate, quite 97% identical nucleotides to the Brazilian isolate. They infect these now subcutaneously in the foot pad, and then they look at the babies, right, that are born. Is that right? They're infected at day 6.5 and 7.5.
Some of them.
Oh, they sack them early.
born.
They sack them early. Yeah, they're each day 13.5 and 15.5.
And they do that because they don't want to have confounding effects of maternal illness on fetal viability.
Yeah.
So they take them before.
What they do interestingly here, a side note, is that the females are lacking both copies of the interferon type 1 receptor gene, and they mate them with wild-type males, so the fetuses are going to be heterozygous, so they'll have an intact interferon response. So
right.
you don't worry about, having a null interferon receptor gene there and the effect that might have. Nevertheless, the virus gets into the fetuses and replicates in them. Some of them die, but they don't see microcephaly in these in utero studies. They do see replication in the fetus. They see placental damage, as we'll see in a moment, but no microcephaly. They have, however, intrauterine growth restriction. What is it? Restriction or retardation?
Restriction.
IUGR. Intrauterine growth restriction.
So he makes the distinction. He says that they didn't see what he calls isolated microcephaly, and he contrasts that with IUGR. So I infer from that isolated microcephaly would be an animal that is normal in size or normal in most ways, but has effects specific to the head.
Mm-hmm.
Right. And so I wind up looking back at the paper that we did that we did just a few moments ago, where they showed that in the SJL mice, is that what they were?
Mm-hmm.
At any rate, that they had intrauterine growth restriction, including effects on the brain, and focused on the brain.
That's right.
That doesn't look to me like isolated microcephaly either. Okay, so I'm just making these distinctions. I mean, if a virus crosses the placenta and just trashes the fetus in a fairly non-specific fashion, that to me is different than microcephaly.
Yep.
Mm-hmm. Mm-hmm. So you're saying that first paper where the mice are small overall, that's not necessarily microcephaly?
That's basically what I'm saying. Yeah, except that I think that one of the things that can happen or might happen, and one of the things that's been recorded in the Zika epidemic in humans even, is intrauterine growth retardation as well as stillbirth. So it doesn't mean that model is not an accurate representation of what goes on. I'm just trying to sort this out and make the distinctions in my mind.
May be that when you put the virus right into the fetal brain, you have a different outcome, right?
Yeah. But in neither of these papers, these are both, one was intravenous, and the other was in the footpad. So they're really looking at crossing the placenta. Right. And in the SJL mice, they got intrauterine growth retardation, but they focused on the brain and called it microcephaly. I mean, they're seeing basically the same sort of thing. They're calling it something different, it seems to me. I guess that's my point.
So did they do any examination of the actual presence of the virus in any of those cells?
In which paper? This one?
Any of them.
Infectious virus?
Any of them. Doesn't matter which paper. I want to know where the virus is first, and then I want to know what it did. So in these mice, this last paper, did they do a body-wide examination for not only the damage that the virus might have done to that tissue, but also the presence of the virus in that tissue?
They, in this paper by Miner et al. With Diamond as the last author, I think this is the one where they say that they can see the virus in the placenta by hybridization.
Yeah, fish, right.
By fish. But they tried looking in the brain, and it's just a mess, and they can't particularly see that.
The brain's a mess?
Well, where can I quote that for you?
So they say here, Dickson, they could see viral RNA in infected placentas, but multiple attempts at fish staining of Zika-infected fetal brains did not yield a clear pattern of viral RNA expression.
But that's not the best way to show that, is it?
Accordingly, we cannot state with certainty whether the enhanced apoptosis in Zika-infected fetuses results from virus or something else, ischemia due to placental insufficiency. Whether fish is good or not, Dickson, they didn't do anything else. But
because you should just do an RNA extraction and look for the virus.
Are you there, Dickson? I mean,
I'm here.
You there, Rich?
Is Richard
can you hear me?
Yeah, I can hear you.
Yeah.
Okay.
Back on the Rich's discussion about them seeing microcephaly or not, this paper by Miner et al. Does do a nice explanation of why they maybe didn't detect microcephaly, and one of the reason is because of the developmental pattern of mice versus humans, and this is one place where there's some difference. And so a newborn pup is relatively immature at postnatal day 1, and so a lot more development has to happen. And so it may be that if you infect the moms at a later time so that the embryos are farther along, then you might actually see some microcephaly, I think was what I took away from that.
I looked back at the other paper that talks about microcephaly, and they did infect them at a later time, I believe.
Mm-hmm.
Right, and it's important to remember, I think, that people are trying to get a generally useful animal model here, and mice are very handy to work with, but of course mice are not humans, and there are going to be differences in the way the disease manifests itself. But this is nonetheless, all of these actually are useful systems, with the exception of maybe injecting the mice intracranially in utero. That seems a little convoluted, but these are all systems. These are all systems where you could start looking at stuff like antivirals and see if you can get an effect.
I'd like to know what's up with these SJL mice. I wonder if we'll ever find that out, because they clearly behave differently than other mice with respect to this phenomenon.
Is anybody curious as to how a mother who's just delivered their pups can distinguish between normal and abnormal pups with regards to this?
How does it happen?
How does that work?
Do you know?
I don't know.
I don't know.
No idea.
Might be based on size, smell.
Probably.
I'm just,
They
smell
could smell death, yeah. What was I going to say? Oh, back on the topic of, Vincent, you mentioned the downregulation of microcephaly genes. I found that counterintuitive, and I checked with my collaborator on that, and she thinks it's just kind of poor phrasing, that they might have phrased it better. Downregulation of genes required to build the brain normally.
Yes.
Yeah.
or misregulation.
Right. You would want to downregulate the microcephaly genes if there were such things.
Right.
So those are altered in kids with microcephaly.
Right.
So presumably they're low expression or they're mutated genes or whatever.
Right.
Yeah. So in this paper, they find clear virus replication in the placenta, and they see good evidence of placental damage. So they feel that the virus is getting in from the maternal blood through the placenta into the fetus. So this is a good model for that. And I don't believe in the Nature paper they actually looked at the placenta, right? If I remember, because those were those.
No, they did not.
Right.
There's one amusing bit in this paper that I just have to point out.
Mm-hmm.
That is, they've got these electron micrographs where they show the virus. I believe this is in placental tissue.
Right.
And in this one where they have this nice little group of 3 viruses, if you look at it, there's this big ugly streak that goes down through the middle of it.
Yep.
And for those who might be familiar with this, that's a knife mark.
Yeah,
Okay, which means there's an imperfection in the knife used to cut the section. And I can just hear the individual who had to probably look around to find these, otherwise they would have used ones that weren't in a knife mark going,
yeah, absolutely.
But there they are.
Yeah, it's a knife mark or a little dull or a chattering or something. Yeah.
Right.
Yeah.
All right. So these are 3 pretty nice models. We'll see how. Oh, one more experiment. Is this the paper? They infected these animals with dengue virus, and there was no crossing into the fetus.
Yeah, I really appreciate the controls.
Yeah,
I think that's good.
So this is really cool that things are moving quickly. And remember, just started talking about this early this year. We didn't know anything, and now we have a mouse model, and we can figure out what's going on. So cool.
We had a Zika discussion in December. We can claim primacy on that.
The first one?
Yeah.
This week, I want to tell you about the annual meeting of the American Society for Virology. This year's meeting is being held in Blacksburg, Virginia, on the campus of Virginia Tech. It's from June 18th through the 22nd. The local organizers are X.J. Meng and Zach Edelman, and their committee are arranging this great venue. The keynote address and plenary symposia were arranged by ASV President Grant McFadden. Keynote address this year will be given by Dr. Stephen Russell on oncolytic virotherapy, followed by 5 days of symposia, workshops, poster sessions, and state-of-the-art lectures. Come listen to Dr. Joe Handelsman talk about leading the White House Office of Science and Technology Policy, be part of a live TWiV audience, enhance your presentation skills at the Career Development Workshop presented by Jean-Luc Dumas. Also this year, ASV is hosting 2 late-breaking Zika virus oral workshops and a poster session. Early registration for the meeting at reduced rates ended May 20th. That's today. Too bad you're going to have to pay more, but you can still register until June 7th online. After that, you go there and register at Virginia Tech. You can go to asv2016.cpe.vt.edu or simply asv.org for more information. Hope to see you all there. Let's do some picks of the week. Dickson, what is your pick?
Well, I have a pick that relates very much to the future of energy use. It appears as though the country of Germany, an entire country, mind you, at a particular moment in time, in this case, I think it was May 10th at 2 p.m. in the morning, declared that they were totally off the fossil fuel energy grid and were using all of the power generated by wind to keep that country alive through the wee hours of the morning until energy demand went up again, and they had to switch back to some fossil fuel. Their goal as a country is to get off the fossil fuel grid in another 20 years, and they better hurry because Sweden is way ahead of them right now, and they might become the first country to ever go off fossil fuels.
Ooh, well, that's neat. 58 gigawatts.
Yeah.
Very cool.
It's amazing how fast. You talk about Zika virus. This is faster than Zika virus in terms of generation of interest in Germany. Doesn't care whether or not, India is still burning tons of fossil fuel from coal or China is burning. We do, of course, but Germany says we're just going to go off. The hell with everybody else. We're just going to go off. They're setting a great example for the rest of the world.
Right.
And this article actually is quite good in talking about the caveats to that.
That's right. It puts everything in context.
This is not. Germany is not fossil fuel free.
No, of course.
This is electricity production, which is
20% of their energy budget is
20% of their energy budget, but it's significant and
very much so
important that they're able to do this.
That's right.
Nice. Well, I put solar panels on my roof, Dickson.
See that? Are they hooked up yet?
Hooked up, but they're not turned on because the town has to come inspect them.
Oh my!
And that takes a
2
while.
more years.
Meanwhile, there's all this beautiful sun that's being wasted on my roof. I can't believe it.
That's the way to look at it. That is exactly how you should look.
You've been wasting that sunlight for years.
I know, but I didn't have solar panels.
Exactly.
Kathy, what do you have?
I picked Entropic Time. It's a YouTube
This is fun.
by Acapella Science. And I can't remember if we've picked one of these before or not, but this particular one is really good because he sings all the lyrics backwards. I mean, to make it, he had to sing all the lyrics backwards. So you can watch the video, and then the next thing that'll pop up will be the making of this video. And then you see how he did it, and it's all the more amazing.
Yes.
It's really good.
Can you sing backwards, Kathy?
No.
Can you sing
I
and
guess
walk
you would
backwards?
you would just practice the words phonetically like you would for singing in a language that you don't sing, but it would be really tough.
Right. That's what he did. He said.
Big time.
Have you ever played records backwards when you had records? You could. The songs are totally different.
Yes.
I mean, totally, like foreign, like from Mars.
Yeah.
Yeah, well, one of those Beatle albums, they say if you play it backwards, it says Paul is dead, right?
Right.
Rich, what do you have?
So recently there was published as a video, a sort of a video op-ed in the New York Times by Errol Morris called Demon in the Freezer. And it's about a topic that we have talked about a couple of times previously, and that is that there are stocks of smallpox in the CDC and in vector in Russia, and whether or not to destroy them. Now, it's fun. I wanted to bring this up, not necessarily to recommend it, because I don't necessarily, but to comment on it because I think people will see it. First of all, I don't know why this is coming up now, because there's nothing new that has happened with this story. Okay. Second, it focuses its attention on two individuals who are on two sides of this issue. One is D.A. Henderson, who was instrumental in the smallpox eradication campaign and wants to get rid of the virus. And the other is Peter Yarling, who's a scientist at NIH, who has been trying to come up with animal models to study smallpox and doesn't feel like that's done yet, and the virus is necessary to do those types of experiments. And so these are two, I would say, interesting characters on both sides of this controversy, and the video is interesting to watch from that point of view. I was disappointed that Morris did not really go a little deeper if he's going to do this at all. And as I said, I'm not quite sure why this is being done now. But I'm a little disappointed that he didn't look at some of the more basic science reasons for.
I think probably the timing and the shallowness of it. My suspicion with any kind of a New York Times feature or op-ed or something by an outside writer is that it's just a good time because that person has something they're about to sell. So Errol Morris may have a new movie coming out or something that this was a convenient promotion for, and I don't know if that movie has anything to do with smallpox. That's just a guess.
So at any rate, people will see this, and I want people to know that although it's interesting and the characters are interesting, it's not the bottom line.
Right.
Yeah, we've talked about this quite a bit, haven't we?
Yeah. And I know I talked to Grant about this because he has his finger on the pulse of this as much as anybody else in terms of the destruction of the smallpox stocks. And there's nothing new going on. Okay? I think it's still kind of on hold.
And I do like Errol Morris, by the way. His work is really cool to watch, but I agree with you. It's a well-made video. Yes, it's a well-made video, but it's not the full discussion.
I wish that the identifications of the people speaking had been more prominent and come up each time. And yeah. I mean, Rich, you know who these guys are, so it was obvious to you. But for me, it's like, now who is this again? And yeah,
they are interesting characters.
My pick is something you've heard about here. We talked about the ASM Augur Art Contest for quite a while, encouraging people to send in submissions. Well, the submissions are closed. We have the finalists all on ASM's Facebook page.
Some of these are amazing.
Beautiful, really beautiful.
Yeah, they're really
creations. And of course, you can vote. You can like which ones you prefer, and you can see there's some clear winners there.
The Monet water lilies.
The Nazca figure.
Yeah, the one with the palm trees is a favorite.
Yes.
The one with there's a cool one with like a double helix made of petri dishes.
Yes.
Chemicals. We got Darth Vader.
The elephant.
Elephant.
It's really intricate.
And of course, we have TWiV.
2.4 thousand. Wow, there's one with 2.4 thousand likes. The little fox, is it? Or some kind of a canid? Yes, then someone made one of me. That's very nice.
That's lovely.
It's with jaundice.
The frying pan egg one has 1.2 thousand. Yeah. 1.4 thousand. Yeah.
The frying. Yeah, that's a cool one. 1.2 thousand. I think the winner must be that foxy thing, right? 2.4K. I didn't realize that. So these are they pick a popular winner, and then there's a judging panel that picks
people's choice.
People's choice.
Well, there's 1.7K.
Really? Which one?
This tree one down toward the bottom.
Oh. Well, you can go there and like them if you wish. They'll be up there for a while. I don't think we judge them by the end of the month, so you have plenty of time. We have a couple listener picks. Matt writes, "Hi, TWiV."
Wait a minute. Did you get? Yeah, did you get Alan?
Nope.
I didn't get Alan?
Oh, you skipped me. I was wondering.
So sorry.
There's a good reason for that. No.
Years, I thought he usually does his last.
There's a very good reason, and you'll find out right now.
All right. Alan, what do you have?
I have a couple of picks. A little while ago, somebody sent me a link to this video called Vertical City, which is a short documentary. I think it's about 15 minutes about this futuristic city design, and they talk to architects and other folks who were involved in envisioning this thing that would take an entire city and essentially put it into one giant, super tall structure. And explaining that, this looks all futuristic, but this could all be done with current technology and the advantages of doing that. And there is, of course, a familiar face who appears in that video.
Despommier.
But what was interesting to me was I saw this video. I thought, yeah, that's pretty cool. It's well shot. It's nicely put together, and it's a neat concept illustration. But at the very same time that I got that link, this was a few months ago, I was in the process of playing through a game on my iPad called Lumino City. If you say it fast, it comes out luminosity, of course. So Lumino City is a gorgeous, gorgeous game. If you have an iPad, you can do it on an iPhone, or I think it's on Android devices. I think it may also be on desktops. This is not a fast reflex shooter type of game. It's a puzzle-solving game. And the whole thing was made... the screens that you're playing on, the scenes that you're going through were actually handmade models.
Wow.
And it's just really, really beautifully done. But it's built, the story unfolds in a futuristic city that was built along kind of these same lines as what they're discussing in the Vertical City documentary. Clearly, this is an idea that's having its moment. Although in Lumino City, the place has kind of fallen on hard times, and your protagonist, who you're navigating through this, needs to put things right, and also to find her grandfather who's disappeared.
It's cool. Very pretty.
The Vertical City documentary is really neat, and I only saw the pic today, so I haven't had a chance to look at the game, but the web page pictures of the game make it look pretty amazing.
It's fun, and as I say, it's a puzzle game, so you don't have to. It's not like hardcore gamers only need apply. It's There are a couple of puzzles in it that were unnecessarily frustrating. It has the worst Morse code interface that I've ever encountered, and I say that with some experience. But other than you get past those, and just the whole story of it is really, really cool.
I'm sorry, Alan. It was a mistake.
That's quite all right.
I just totally forgot. I will do one listener pick in the interest of time, and because Rich is here, we have a pick from Jenny, who sent this especially for him. Are you there, Rich?
I'm here.
Hi, everyone. In TWiV 384, Rich picked Dune, which I agree is among the best sci-fi novels of all time, and said he's always looking for new sci-fi. I'm here to help. I have a few recommendations for you, Rich. Ancillary Justice and its sequels by Anne Leckie. This is a must-read. Ancillary Justice is the only novel to win the Hugo, Nebula, and Arthur C. Clarke Awards. It's spectacular. It has clones and sentient machines and gender politics, and is brilliantly written. You should be reading it right now.
Actually, I just did read that, so thank you.
Good.
And I'll look at its sequels.
Parable of the Sower by Octavia Butler, published in 1993. Butler was one of the great American sci-fi writers, but is generally underread because of her race and gender. I highly recommend all of her books, but this is my favorite. You read that one, Rich?
No, none of these others have I read, and I really appreciate this. This is great.
The Fifth Season by N.K. Jemisin is one of the best books I read last year. It's not typical spaceship sci-fi, but it's not swords and sorcerers either. Jemisin is another author underread because of her race and gender, but this is her best book to date. The plot twists will rip your heart out. God's War by Cameron Hurley. This is not for everyone. Hurley pulls no punches on gender, violence, and individual monstrosity. The writing is not quite as polished as her more recent books, which are more fantasy than sci-fi, and I don't know if you read that genre, but if you want to go for a hell of a ride, Hurley will take you there. And then The Passage and its sequels by Justin Cronin. If you like government-created vampires, this series is for you. Much better than your average novel about government-created vampires. Wow, she writes really nice reviews.
I really appreciate this. This is great because I'm always looking for reads like this. This is great.
You need your heart ripped out, Dickson?
It's gone already. I gave it to Marlene.
Oh.
Oh, shucks.
Shucks. That's it for TWiV 390. You can find it at iTunes, at microbe.tv/TWiV, at Google Play Music, Stitcher Radio, and all good places that you can grab podcasts with your favorite podcatcher app for iOS or Android. And send us your questions and comments to TWiV at microbe.tv. Dickson Despommier is at verticalfarm.com. Thank you, Dickson.
Welcome, Vincent.
Did you have a good time today?
Had a wonderful. Except for my reading skills, I have to sharpen them up.
That's all right. Don't worry about it.
I angst over things like that. Sometimes I read well and sometimes I don't.
Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This was a good time.
Rich Condit is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure enough, always a good time.
And you're going somewhere now? You leave? Oh, you're going to Penn State, right?
Yeah, I'm going to go see you.
I'll see you next night. You're going to go to the meeting. I'll see you there.
Yeah.
Okay. This should be fun.
Yep.
Alan Dove is at alandove.com. You can also find him as Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I didn't forget you.
No, you didn't. Are you guys going to Penn State Hershey?
No, the
Penn
other
State.
one.
Oh, the other one. The non-chocolate one. Okay.
What's
Belvin.
the name of the town where the other one is in?
State College.
I think.
State
And
College.
University
State
Park.
College.
And
specifically.
University Park,
Yeah,
yes.
and we get to stay at the Nittany Lion Inn, and we get to go eat ice cream at the Creamery. It's all good.
Oh, nice.
All good.
There's a lot of recent bad press on Joe Paterno, so be prepared.
Yeah, no. Rich is giving a talk called The Accidental Virologist.
Right. This is the
Nice.
this is the old retired guy's wisdom talk.
Well, I'm not
Cool.
giving any talk.
So are you doing a TWiV
We're
out
doing a TWiV with Forrest Rower
Nice.
and David Price. Should be good. Well, it's the meeting's focus is the virome, and
It looks like a great meeting.
Should
Really
be good.
nicely organized.
yeah, it should be good.
there? When is that?
Starts Monday and goes through Thursday.
Oh, okay.
You could go, Alan.
What day are you going to be TWiVing out there?
Thursday night.
Thursday
No,
night.
Come.
No.
What?
Not Thursday night, because I'm out of there. I think it must be Wednesday night.
No, it's Thursday night.
God, it better not be. 'cause I
Uh-oh.
won't be there.
Dude, you messed up. Well, let's talk about it.
You'll have to sort that out. Okay. Never mind.
Anyway, I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.